Good morning. We are going to discuss gene regulation today. This is a little more complicated. As a general rule, I didn't really go over this in biology when I taught it, just because it is so complex and you, quite frankly, won't read about or need to know about gene regulation until you get to college. And even then, just depending on what kind of a biology class you take, um, you may or may not even discuss gene regulation. So this is a little more complex. I do have a couple of diagrams to help understand, so hang in there, we'll get to the diagrams. Okay, kind of talking about protein synthesis and making proteins. You're going to have two different ways that genes will be expressed depending on if you're a prokaryote or eukaryotic cell. Obviously, eukaryotes are going to have way more genes than a prokaryotic cell will have. They're more complex, tend to be a part of multicellular organisms, and so they do need more genes. Both types of cells are going to be able to regulate and control what types of genes and when those genes will be expressed. Obviously, it's going to depend on when the cell needs certain proteins or if it needs a certain protein. Sometimes it'll depend on environmental factors. On average, your cells will only express about 3 to 5% of their genes at one time. Different cells will express different genes. Your skin cells don't need to express your eye color. So we're going to get started with the gene expression in prokaryotes. So in bacteria, you have this, one of the more well-known ways of gene expression is going to be the lac operon. The lac operon is very specific to lactose, but an operon is just a group of genes that code for the cell to make certain enzymes that will do, obviously, a certain job. All operons will include a promoter, kind of where it starts in the operator. Promoter is going to be a couple of nucleotides at the very beginning. The operon will be another set of nucleotides right after the promoter. Like I said, the lac operon is the most well-known of the operons, and its job is to break down lactose when it's present in your cell. So E. coli, for example, has the ability to break down lactose, the milk sugar. So that's why we have E. coli in our intestines, because our cells can't break it down, but the E. coli can. They can break it down into galactose and glucose, which our body can then metabolize from there. The lac operon has 3 enzymes that come right after the promoter and the operator. Each enzyme is made by its own different gene. So they're all right next to each other. I know this sounds really abstract, I promise. When you see the picture, it should make more sense. So like I said, it's a series of bases. You've got your promoter, then your operator, and then the 3 different genes. And so the promoter ends up controlling all 3 genes. It's going to determine when all 3 of those get expressed, and they all get expressed at the same time. So its job is to just determine when we're going to make these proteins. When are we going to make these enzymes? The operator's job is the on and off switch. So promoter tells the operator when to turn on and off. Specifically, the operator controls when RNA polymerase can attach to the DNA strand and start to, the RNA. The RNA polymerase can attach to the DNA strand to start transcribing. And then obviously, translation. So, when there is no lactose present, there's no point in making these enzymes, and so the operon's turned off by a repressor protein. It basically sits on the operator, and because it's sitting on the operator, RNA polymerase can't attach, and so you physically cannot transcribe the genes behind it. When lactose is present, it tends to bind to, well, it doesn't tend, it does bind to that repressor protein, so then the repressor protein doesn't have the ability then to attach to the operator. So that's how prokaryotes regulate when their genes are going to be expressed and not expressed. So here's the picture I was talking about, okay? So here's the lac operon right here. Here's your promoter. That's a set of bases right there. And this is where RNA polymerase will attach to start the transcription. You can kind of see here, this does a really good job here. This is the operator. This little piece right here is the operator. This little guy right here is the repressor protein. You can see how it's going to attach to the operator. On this other side right here, this little triangle looking thingy, maybe it's a half moon, half of a half moon. You can see how this fits in nicely to the operator right here. So it literally sits right there. So even if the RNA polymerase attached to the promoter, it physically can't get around the repressor protein when it's attached to the operator. And so then it can't transcribe the rest of these genes back here. So gene 1, 2, and 3. Then what that little notch there is, if there is lactose present, it's going to attach here, and so then it's preoccupied, if you will, and then RNA polymerase can still attach to the promoter, but there's nothing in the way, so then it's able to go ahead and transcribe. So hopefully now that makes a little bit more sense. Okay, eukaryotes. This is a little, I think it was a little more complex for me to understand when I was learning this, but they use transcription factors, which is just a fancy word for protein, a specific protein, to regulate when the genes will be expressed in eukaryotes. So what the transcription factors do is they help arrange the RNA polymerase onto the promoter, kind of directs it to attach to the promoter so the RNA can attach to the DNA and start transcription. There is an enhancer. This is a piece of DNA that's not actually attached to the gene, so it's like there's an enhancer, a little bit of DNA in between, then you get to the promoter. I've got a picture, don't worry. So when that enhancer is able to bind with your, it does bind with the activator, which is a special transcription factor or a special protein. So basically an activator is a protein. So the enhancer is going to bind with the protein, which is the activator. So the activator is a specific transcription factor, which is a protein. I know it sounds really crazy. It's a lot of words. So when you have the now activator enhancer, because they're now attached to each other, when that comes in contact with the RNA polymerase that's on the promoter, then transcription can happen. You might be thinking, how is that possible when the promoter with the RNA polymerase is not attached to the enhancer? There's a space in between. That's because DNA is going to fold so that it can join together. Let me show you a picture. Maybe. There, oh, just kidding. There we go. So here's your enhancer, right? This is the section of DNA that's not attached to your gene back here, which is going to be transcribed. The activator, which is the specific type of transcription factor or protein, attaches to your enhancer. There it is right here. Okay. The RNA polymerase binds to the promoter over here. So that's RNA polymerase back here. Here's another transcription factor. So the protein there. So what happens is this binds to this at the very end here. And then this is what I mean by the DNA folding, so that the enhancer and the activator are now in contact with the RNA polymerase who's bound to the promoter, and then transcription starts. So you might need a little bit more time on this one. So I'll come back here and then you can pause this. I'm not sure what just happened. I started to go backwards. Okay. Okay, last slide. So after your cell goes through cell division, normally your DNA is not going to be in chromosome form, which is a very tightly wound X shape. It's only in that chromosome X shape when it's ready to go through cell division. So normally it's relaxed. It's in the pieces of chromatin, which are just the relaxed pieces of DNA. Chromosome is the duplicated DNA. So the left side matches the right side. So when you have your little X shape here, the left side of the X here and the right side of the X, those are two copies. They're exactly the same, and then they're held together by that centromere in the middle. The euchromatin is going to be the portion of the DNA that is uncoiled in the cell. It's going to be the part that, since it isn't all wound together, all the different proteins can get in to do the transcription. So this is the part that euchromatin is the good part that's going to be able to be transcribed. Now remember, we're in eukaryotes, so we've got those introns and exons. Some portions of the DNA never uncoil and so will never be expressed in that cell. And because transcription and translation occur in separate locations, eukaryotes are going to have more control over what genes are going to be expressed. They're going to be able to take that little step in between and cut out snippets that they don't want expressed. So, if you have questions, which you might, just ask. This is, like I said, this is a more complex set of notes. So, if you are overwhelmed or need a little bit more explanation, please don't hesitate to ask.